Welcome to The Explainer. Today, we're skipping the dry textbook biology lesson and diving straight into a masterclass in natural engineering. We're talking about plant reproduction, or, as I like to call it, the ultimate biological assembly line. I mean, really think about it for a second. How do stationary plants manage to reproduce, find mates, and literally conquer the globe without ever taking a single step? It's a massive, mind-boggling logistical problem. But, as we're going to see, evolution has brilliantly solved it. Here's the roadmap for today. First, the anatomy of a flower. Then, the journey of pollen. After that, fertilization. Next, we'll crack into fleshy and dry fruits. And finally, we'll wrap up with germination.
All right, part 1, the anatomy of a flower, the master factory.
So here is your master factory. You'll notice everything sits right on this swollen base at the bottom. That's called the thalamus. Now check out the two main reproductive players here. You've got the male androecium, with its long, pollen-producing stamens, and right smack in the center is the female gynoecium. That one features a sticky stigma at the top and an ovary down at the base, which holds the ovules.
But hey, not all factories are built the exact same way, right? A complete flower, like a China rose, is fully equipped with both the male and female parts, the whole shebang. On the flip side, an incomplete flower, like a papaya, is unisexual. It only has one or the other.
Moving on to part 2, the journey of pollen, solving the logistics.
If you literally can't walk over to your mate, you're going to need a delivery service. And that is pollination. By definition, it's just the transfer of pollen grains from the anther to the stigma of a flower. It's the plant world's elegant, fundamental solution to the whole mobility problem.
Now, this delivery can basically happen in one of two ways. Self-pollination is essentially an inside job. The pollen transfers to the stigma of the exact same flower, or maybe another flower on that same plant. Cross-pollination, though, that's when pollen gets transferred to the stigma of a completely different plant of the same species.
And this is where we see form beautifully following function. Look at the maize plant. It relies entirely on the wind, right? So it completely skips the showy colors and just churns out massive amounts of super light pollen. But then compare that to the hibiscus. Since it relies on insects, it acts like this giant brightly colored, sweetly scented billboard. It produces sugary nectar just to attract delivery agents like bees and butterflies, who then inadvertently carry that sticky pollen right on their bodies from plant to plant.
Which brings us to part 3, fertilization, making the seed, the epic microjourney.
Okay, so once that pollen successfully lands on the sticky stigma of a compatible plant, the real quest kicks off. It doesn't just fall in. The pollen grain actually germinates right there on top and sends out this tiny microscopic tube. And this pollen tube physically grows downwards. It tunnels entirely through that long tube-like style, carrying the male gamete until it finally reaches the ovule, nestled safely inside the ovary. It's honestly like a highly targeted, microscopic drill.
And that brings us to the absolute crucial moment, fertilization. This is the exact fusion of those male and female gametes to form a single-celled structure called a zygote. This tiny little zygote, it is the literal beginning of the next generation, perfectly destined to divide and eventually develop into an embryo.
On to part 4, fleshy and dry fruits, the protective packaging.
What happens immediately after fertilization is actually super dramatic. The flower's primary job is officially done, so its petals, sepals, and stamens usually just wither, shrink up, and fall off. But the fertilized ovule, that hardens to become the seed, and the ovary surrounding it, it enlarges, swells up, and actually becomes the fruit. So yes, the fruits and seeds we eat are, biologically speaking, just protective packaging designed to keep that embryo safe and help it get dispersed.
Now, these fruits generally fall into two main camps. In dry fruits, think of a garden pea or a walnut, the entire outer wall of the ovary, which is called the pericarp, becomes thin, dry, or even as hard as a shell when it matures. But in fleshy fruits, that pericarp develops into the succulent, juicy tissues we love to eat.
Let's use a mango to see exactly how this builds out. The pericarp of a fleshy fruit actually has 3 distinct layers. That thin outer protective skin? That's the epicarp. The delicious, pulpy, edible part in the middle is the mesocarp. And finally, that really hard, membranous inner layer protecting the seed in the center is the endocarp. It is just a brilliant, multi-layered biological defense mechanism.
And finally, part 5, germination, a new beginning, the starter kit and activation sequence.
If we were to crack open a fully formed seed, we'd find a ready-to-go starter kit. On the outside, you've got the seed coat, basically tough outer armor. But notice this tiny little hole called the micropyle. That is a totally crucial design feature. It acts like a doorway, allowing water to enter and wake up the dormant embryo sleeping inside. And that embryo has two primary parts: the radicle, which is going to become the root, and the plumule, which is destined to be the shoot.
But here's the catch. The embryo needs fuel to start growing way before it can actually photosynthesize. So the seed basically packs a lunch. And this comes in two distinct varieties. Monocot seeds, like maize or rice, have just one cotyledon, or seed leaf, and a separate tissue called the endosperm that holds the food. Dicot seeds, on the other hand, like peas and beans, have two bulky cotyledons that store all the nutrients that little embryo needs to get going.
Now, even with its food all packed and ready, the seed won't sprout just anywhere. It requires a very strict environmental activation sequence. First up, it needs adequate water to soften the coat and crack the seed open. Second, it needs oxygen to release energy from its stored food through respiration. And third, it needs a suitable, optimum temperature. Without all three of these specific conditions being met, the plant just stays asleep.
But when those conditions perfectly align, boom, we get germination. This is the miraculous process where that dormant, waiting embryo finally wakes up and develops into a young plant or seedling. The life cycle officially begins anew.
So once water is absorbed, the seed swells right up and the coat cracks. The radicle is always the very first part to emerge. It shoots straight downward into the soil to establish the root system and firmly anchor the plant. Right after that, the plumule stretches upwards, breaking through the surface of the soil to bear the plant's very first leaves.
So as we wrap up today's explainer, I really want to leave you with this thought. A tiny, seemingly lifeless, completely dry seed can literally wait out there in the dirt for months, sometimes even years, completely self-sufficient. It patiently holds on to its protective packaging and its packed lunch until the exact perfect moment of water, oxygen, and temperature arrives so it can burst back into life. I mean, doesn't that make the simple seed the ultimate biological survivor?